what I'd like to talk to you, and I'm recording this so that I can send it out to the couple people that are absent today. I've gone over it with the other kids in all the other classes. I have gone over it with all the classes. Let's go through this and make sure you understand what's going on. I'm talking about the lab that you were supposed to do yesterday. You were given a video a week ago to watch for Punnett Square, not by me. It was made by Bozeman, another AP teacher from another city. I don't even know where he is from. But it's a good video and people like it, so I sent it to you. Yesterday or day before, I can't remember when, it was day before, I sent you the video explaining how to do chi-square in detail. Again, not done by me, right? And a video that's required to do this lab. In fact, it says number 1, procedure. Number 1, watch the short film, The Making of the Fittest, Natural Selection of Humans. So you had to watch that. If you haven't watched that, well, you're behind. You got to figure it out. But you have to know what you're doing. And I got no time to have the discussion with you about it. I will be here after school till 4 today, till 6 on Friday, and I will be here on Saturday school. But we're not going to spend class time on that. That's stuff that you're expected to have done already. That was homework. That was classwork. If you missed class because you were dancing or you were on a field trip, then you should have done that stuff for homework. That's the expectation. If you're not meeting that expectation, then you failed yourself. Not me, you failed yourself. All right, let's go ahead and move on. You have, in the language and the introduction, they give you some explanations, and it's really important that you read the introduction and that you understand the introduction. In the introduction, they say HBA. HBA is the same as homozygous dominant in this case. Homozygous, in this case, HBA means normal hemoglobin. They also write the term HBS. Now, they explain it. They don't expect you to know it. They tell you. But I've had many students come and say, what does this mean? I said, well, read it. It tells you in the paragraph. Too many kids want me to read it to you. I'm not going to do that. HBS. It says, HBS says you have homozygous recessive. In this case, of course, we're talking about sickle cell, so that means you have the disease, so that's your recessive. And when they say heterozygous for HBA, and do you understand that we could say heterozygous for HBA or we could say heterozygous for HBS? Do you see why we could say either one and then the same? No. Then you need to study, do some more studying. So that means heterozygote. So that means both. So in the case of symbols, if we went by symbols, this is big S, this is little s, and this is big S, little s. So HBA is two dominant traits. HBS is 2 recessive traits. Heterozygous for HBA means you have one big S and then the other something else. If I say heterozygous for HBS, that means I have a little s and I have something else. So they're the same thing. If you don't get that, again, it's information that we've gone over repeatedly in many different example problems long before, since January, since the first day in January that we came back from break, and now we're ending February already. So at this point, wow, you should be feeling a lump in your throat. You need to feel like you need to get going. On page 2 of 12, you see it says questions, Mendelian genetics and probability. Page 2 of 12, then there's 3 of 12, then there's 4 of 12. Then there's 5 of 12, then there's 6 of 12, then there's 7 of 12, and then there's 8 of 12. That should be 100% done at this point. My expectation is that those pages 1 through 8 are complete. And you know what? My expectation is that they're 100% correct. Why do I have that expectation? Can someone answer that? Why would I expect, and I'm just going to use the word good student, right? Why would I expect that a good student right now, I should be able, why would I think that I should be able to get up right now, go around this room and grade pages 1 through 8? If you have even one wrong, make an effort, the whole thing. Why? Why should I be able to do that? Should have done it already, and what other part of that? Why do I think you should have got it right? Yeah. It's in your Jupiter grades, and if you don't have one here, somebody's taking more than one. I don't have time to handle it, so you have to deal with it. Whatever, you can figure it out. I gotta get moving. I'm moving on. So why do I think that pages 1 through 8 you'd be able to get done? 100% accuracy.
You had the video.
No, not because of video.
You just read it.
No. Have you even looked at it?
No.
Yeah, I know you haven't, dear.
You had it yesterday.
You had it yesterday? What did you notice about it? Punnett Square, probabilities, dominant recessive, pedigrees, because you already know it. You've been tested on it. You've had probably 7 hours of video and homework and reading and tests and whole genetics tests that I told you had 20% chance of getting a boost on if you came after school. By the way, not a lot of people taking advantage of that. Still waiting. All that's been done. So my expectation of a good student is that you already know all that. So pages 1 through 8 should have been done, and it should be 100%. No one should have a problem doing a Punnett Square. Nobody should have a problem answering. This is stuff that's going to be on both those tests that are coming up. Both in March and in April. You're going to have standardized exams. They're going to ask you these questions. We've been hitting it since January. I've had tutoring hours late into the night with many people. If you can't get this right, you better figure it out. All right, so number 9 is new material. Number 9, page 9, number 11 is the one we're going to do today. So number 11 asks you to figure out, well, it's asking you to figure out, is natural selection happening or not? So it reads, the multiple couples living in a small village in the Eastern African lowlands. If you have not watched the video, which my understanding is based on some responses in the class, you haven't, well then you're in trouble. Because you're not going to be able to understand this as deeply as if you would have. It's okay that you didn't read it. I'm not grading it. I'm not putting a grade in the grade book. But you're going to have, it's not okay because you're going to get a bad grade on a quiz and a test. And you may not graduate high school if you keep up the same behavior. Is that clear? You have to do this stuff before you come in. You have to do your assignments. You're given an assignment, you got to do it. So anyways, and that's the first step in the procedure. 1 through 8 should be done. Let's go ahead and start with number 11. So in Eastern African lowlands, all of whom have heterozygous for HBS. What does that mean? Heterozygous for HBS. What does that mean? Anybody? Big S, little s. That means everybody's big S, little s, and everybody's having babies. What does that allow me to do? Automatically, my brain goes right to doing a Punnett Square. Right away, I know my percentages. I know I got this going on. I know that I got this going on. I know that I got this going on. I know that 25% are little s, 25% are big S, and 50% is big S, little s. I know that. You know that. If you don't know that, again, you're in trouble. You're in trouble. You need to repair that. And here we're talking about percentages. That's it. That's important that I know that. If you don't know that, it's going to be hard for you to understand anything else. Which is why I've been hitting it so hard over these last 2 months. All right, so in the problem set, it says, look, you have 500 children. 500 children. Not a person. We're talking about for a village.
Oh.
500 kids in a village.
Oh.
Not a person, yeah. These aren't rats. These are human beings. So 500, rats can have 500 kids, right? Human beings can't have five in a lifetime. You're not going to be able to have 500 kids, right? So that's why I said these are not rats. These are humans. Say what now? Huh? Wow. Okay, whatever. Yes, that's possible. You're right. Whatever. All right, so this small village, 500 kids. Now, out of that 500, what does the 500 represent? Somebody said it. Say it loud. So I? Total. That's right. It's the total. This is important. You saw that in Hardy-Weinberg. The total is important because that's what's going to let you get the proportions. All right, so you have 139 of the kids are homozygous for HBA, which means what? 139 are what? Homozygous dominant. Big S, big S. You notice what I did? I wrote it all down. I wrote down what I knew. Do you see that? First step in problem solving, read. And read for understanding. Second step in problem solving is writing down what you know. That's why I wrote this first, and then I wrote this second. Because those are two things I know. Yes. You have to speak up. Yeah, these are what are observed. That's a good point. What you're seeing here is when somebody tells you, of these children, this is what happened, that's what happened. That's what you observed. All right? Flip to number 12 real quick. Flip the page over to page 10. Look, where are the observed there? Look at number 12. Where are they observed? Number of children for blood test results. The test results. So it's what you observed is, it depends on the problem, what they name it, right? So 139 homozygous dominant, 279 are what?
Recessive.
Are heterozygous, please read, big S, little s, and 82 are what? This says 82 suffer from sickle cell. So they have to be little s, little s. Good. See, you know. You just got to use the brain. So you got little s, the ones that have the disease, right? Just like in Hardy-Weinberg, if it's a recessive trait, if the person has the phenotype, then you know the genotype, right? So here you have 279, 139, 82. Are we clear? Okay, so what can you fill in? You can fill in one third, a little more than 1/3 of that chart. What can you fill in? Phenotype genotype, and what? Observed. Fill it in now. Get it done. I just did it right here. Genotype big S, that's HBA, homozygous dominant. 279, which is what? Big S, little s, or heterozygous for HBA, hetero, big S, little s, and 82 is the little s. Those, the phenotype is they have sickle cell disease. The genotype is little s. Or another way you could write the phenotype is to say they're HBS. Are we good? Actually, these three are the genotypes as well. Just another way of saying genotype. All right, good. So this is normal, big S. This is normal hemoglobin, big S with the trait, big S, little s. And this is disease, little s. Are we good? All right. I'm not going to say. We got to move on. I'm sorry. Don't mean any offense, but you can come see me after school. All right. We just got a lot to do. We're not going to have time to do it. I'm sorry. All right. So that gives you what? What did we say this was? Observed. Very good. This is your observed. What's the other thing that we, according to that chart you see there, what's the next thing we need to do? The expected. Now this is the part that most people get really confused on. Because the observed is, I hate to say this, but the observed is what they tell you in the problem, right? Sometimes it's going to be hard for you to find it because they're not going to use the word observed. They're going to use test results, or they're going to say you have 500 kids, and you got 30 of each of these kids. So the observed, they're gonna tell you that information. Is that clear? But the expected, you're gonna have to calculate. So that's gonna be a little more difficult. Not by much, though. Not by much. So you have the big S, that has to equal something. I don't know what the expected is. Big S, little s, that has to equal a certain number. Who knows what that is? I don't right now, because I'm still figuring out the problem. Then little s, that's a number. So how do you find these numbers? Go ahead.
Let's just divide, say the big S is equal to 139 divided by 500.
No, that would give you the probability, that would give you a percentage of how many you have that are big S. That doesn't give you expected. The question for expected is how many do you expect to see? This is how many you see. How many do you expect to see in the population? Can anybody tell me before I explain to you how you would find the expected number of kids that have one of each of these different genotypes/phenotypes? Yeah.
Would you get the percentage of each one and then multiply it by
How would you find the percentage of each one?
139 over
No.
500?
That's telling you listen to what you're saying. Listen to what you're saying. I'm asking you, what percentage, how do you find the percentage you expect to see? You're telling me the percentage of that are. You're telling me the percentage that you see. I'm asking you, what's the percentage of each of these that you expect to see? Where in this problem, I'll give you a hint, the answer's on the board. Where do you see, how do you find the number of individuals that you expect to have big S, little s, and little s. Say what? Punnett Square. You see, I knew you could figure it out. I knew it was in your head, right? It was, right? I didn't tell you. It was in your head. I knew you knew that. And there's the answer. There's the answer. It's right here. You did it right here.
Oh.
You already did it. That's why you do a Punnett. Why do you do a Punnett Square? Not for fun, although I find it's fun. I like it. But the reason you do this is so you know what percentage you expect to see. That's what a Punnett Square is. What do you expect to see? It's never what it is. Well, rarely it's what it is, right? It's what you expect to see. Your observed are what? What it is. All right. So of the big S, what percentage do I expect to see?
25.
Not 25. It says 20. It's 25. Now listen, it's 25%. You're right. Of what? 500. What does 500 mean? What does of mean? Times. So it's 0.25 times 500. Why 500? That's your total. When I say you got 25% of the people are big S, that's 25% time, or 0.25 times the number of people.
It's 125.
What is it? 125 is correct. So that's 125. Now, if big S, little s, I expect to see what? 50%. And what's that? I shouldn't have that. You shouldn't even need a calculator for that.
250.
Yeah, thank you. It's already written on. Appreciate it. So little s is what? 125, because it's the same percentage, right? Now, isn't that easy?
Yeah.
So there's your expected. You already got your observed. It was given to you. So that chart, the observed and expected, boom, you're done. By the way, that's little e for expected. And this is little o for observed in the formula when we get there. Now y'all have that written down because I'm going to need it when we go on, and I'm going to change the slide. Is that okay? Y'all have it written down? I may need you to tell me those numbers. All right, so before we could go on and try to figure out what is chi-square, what is critical value, what does the table mean, what is degrees of freedom, all that depends on you understanding what a normal curve is. So let's just quickly go through that. So here you have a bell curve. You're all familiar with it. We looked at it in grades when we did grades. When you look at a bell curve, there's an area underneath the curve. This area right here, this area over here, that's not that's not close to not close to the mean, right? This is kind of, if you look at it, if you think about it, this part here, this is where what? What is the white part for? What does it tell you? What does the white area tell you?
That's the average.
That's good, close. The average is actually right in the center, right? So the mean is actually going to be right here, right? Right? So what you have in here is that this is where, there you go, good. This is where you, perfectly said, by the way. Good job, Curry. Is where most fall, right? If we looked at weight or height or we looked at grades or whatever, you expect to see in a normal distribution that you get most of the people, most of the events happen in here, right? And you expect that the least number of events happen here or here. Do you agree? Now I could fill them in any way, and this is the number of events. Now I can fill this in any number of ways. I could say, okay, well maybe here's the F's, here's the A's, and here's the C's, right? This is a lot of people like to think of it, but you know what? It doesn't have to be that. It could be these are the B's, these are the A's, and these are the A pluses. I always love when that happens, right? That could happen too. But all we know for sure is that if it's in this area here or this area here, those are the few. There's only going to be a few that have F's. There's only going to be a few that have A's. Does that make sense? Everybody, or there's only going to be a few that have B's, but there's always going to be a few that have A plus. You know what? It might not even be that. It could be A minus A and A plus. Right? So the numbers, what my point is that these values change, but guess what always stays the same? This distribution of, this is what you expect if things are just random, right? You'd expect to see it this kind of, if there's no real relationship, nothing changing that. You'd expect to see, in the case of grades, some kids doing really well because whatever reasons, and some kids not doing that well, and some kids doing what we call the average, whatever that average is. Are we okay with that so far? Yeah. What a great question. Yes, it's weird, but I mean, I don't know about upside down, but the curve can change. But it's weird. I don't want to talk about details because we're just going to try to get the basics done. What's going to be on that test? So when you take it, and we could talk about it after class, by the way, if you want. All right, so you have a graph, and what chi-square, the graph for chi-square looks like this. All right? It's a one tail. It's one tail, not two. By the way, you know what I mean by tail? Do you see what I mean by tail? Make sure you understand. What I mean by tail is these red areas. This is a tail, and then this is a tail, right? See how it looks like a tail? This looks like a dinosaur, and this is like hangs out this way. There's two tails. I mean, I see this like a brontosaurus laying on its side, and there's a tail this way, tail this way, but whatever. I don't care. If you don't have any imagination, it's your problem. All right, so anyways, here we only have one tail. Do you see that? So we call this one tail kind of graph here. And then before I go on, because this has been a problem, this is a weird problem for me because I wasn't expecting. It's the first time I had someone ask this question. So some people don't understand that this is the lower number, and then it gets higher as we go this way. Do you understand that? That this is kind of it doesn't have to be a zero, but it does have to be lower than this area. It's always getting higher going to the right. Right? It's a number line. Right? Two number lines, a Y and an X. Do you see that? Okay. These are still the number of events, right? These are still the number of events that fall in a certain range, whatever that range is. We don't really know. Whatever the X is, it doesn't matter. That's not the point. Now this red part here, the area that's there, we're going to call that the probability. And we're going to say, not probability, we're going to say P or alpha, okay? That's all the probability within this red area, All right, and it turns out that you can set that probability to whatever you want. Please take a look at the chart on page 12. Please take a look at the chart on page 12. What are the probabilities that they have on that chart? By the way, they have whole books of this stuff. They have to, if they want you to calculate the critical, if they want you to find the critical value, they're gonna have to give you this chart. Are we clear? All right. There are other ways of finding it, but this is, at your level, what we're talking about, you need to be expected to get given this chart. Yeah. You come and ask me a question? You gotta go to the restroom, go. What's going on?
I wasn't here for the last 2 days,
Uh-huh.
so I never got that. I never had to pay.
Well, you should have asked before. Is there any, there are no other examples? Did you not check your Jupyter grades and print a copy? You didn't do that? I don't know what else to say. Try to share with someone next to you. I don't have any more copies. All right, so anyways, Zaid, why are you up here? Lord Jesus, give me strength. All right. So here we're talking about the area or p-value. Where on that chart do you see? If you, son, you don't have a copy of this. So you need to sit next to someone you can see the chart. You have to see these things. I'm sorry I don't have a copy. I can print one for you after class, or you can print it for yourself. Guys, you're absent. You got to print your stuff. I know you don't have printers at home, but there's printers in the hallway. I see people printing stuff all day long. Get it printed.
We're still looking at this chart. Why you
Yeah, you're looking at this chart. On that chart, where do you see P? Top left. So that's that whole top portion are p-values. So anywhere from 0.995 all the way down to 0.01. The smaller that p-value is, the smaller this area is. This area is known as the rejection area. Or a zone, or there's all kinds of names for it. But it's the area where you reject the null hypothesis. Oops, there's something that you've heard before, but I bet you a lot of you forgot. It's the null hypothesis, and the null hypothesis is the hypothesis that says it's just random. There's nothing is connected. There's no relationship. Whatever that relationship is, okay? In the case of the example in the video, dice, right? So if so null hypothesis, in this case, in this example, the problem we're doing, the question is the distribution of the genes, is that due to random luck? If it is, it's the null hypothesis. The null hypothesis is when the observed is due to luck, right? Random chance. Do you understand that?
Yeah.
Okay. So that means it's nothing, there's no relationship between, in the case of the example that they gave in the video, it was about whether the dice were fixed, were they fair, or were they unfair? All right, unfair means somebody's cheating, right? But in the case of this gene distribution, is the gene distribution random, or the alternative hypothesis? What's the alternative hypothesis, which is opposite of the null hypothesis? It's not due to luck, it's due to natural selection. So an alternative hypothesis is not going to be due to luck. It's not random. There's something going on. There's something's changing the results, right, from the expected. The difference between the expected and the observed is so big, it can only be due to something else happening. Is that clear? So I got 5 people that in this room that chew gum. God forbid they all have cancer. They all get cancer. Is it random that they all got cancer, or is it because of the gum? Well, you could say that, but you really need to do what? Research and test it, right? Yeah.
Can they be biased?
Can what be biased?
The observations and stuff?
Absolutely. There's all kinds of error that can happen. All right. So bias is one of the big errors. So for sure, right? But right now we're just talking about how do you use chi-square. And so chi-square, so this is what we know. We know the relationship that we have a null hypothesis and we have a random hypothesis, and that we got and then we got an area under the curve that's equal to p, that one tail. That p is the probability, and it's usually, let me tell you this, that usually we set it, usually the value usually is equal to 0.05. In the video they set it to 0.01. The smaller this p-value, the smaller the probability, the more likely it is that if your chi-square test is bigger than your critical value, the more likely it is that you're right, that there is a relationship. Remember, you can always be wrong, right? We can't prove anything in science. That's why we use the null hypothesis, because we can't prove it, right? We agree, we can't prove it. So all we're trying to do is disprove it. Disprove something. We're trying to disprove the null hypothesis. If we disprove the null hypothesis, then we accept the alternate hypothesis. We accept that something's going on. Does that make sense? Okay, yeah.
Can there ever be an in-between the null hypothesis and the alternative hypothesis?
Yeah, there's all kinds of things that can happen, yeah. For this example, for what we're doing, it's either one or the other. And that's with every experiment. You know, just because we say that there's an alternative hypothesis, we say there's natural selection going on, is that necessarily true? No, we need to do more investigations, more experiments. We need to collect more data. We need to make observations. We need to figure out what the expected values are. We have to compare the expected to the observed, and we have to decide, is there a relationship or not? And it has to keep going over and over and over and over again, right? Does that make sense? It's only till we collect all libraries full of data and studies and chi-square tests, and only when we have libraries full of data do we start to say, okay, we think we're pretty sure we know what's going on here. All right, so prediction area. Now, there's another word that was used, and it's a real special word. I'm gonna put it in yellow. And there's a number here. There's a number here that's called what? Anybody know? Critical value. All right, the critical value is a number that you must know. You must know the critical value. How do you find the critical value? To find the critical value, you have to know one other thing, and that's the degrees of freedom. The degrees of freedom is easy. The degrees of freedom is something that is a real simple relationship. It's n minus 1. It's the total number of possibilities minus 1. So for this problem, for problem number 11, what would be the degrees of freedom? What are the possibilities for this problem?
What did we 138? Hold on. Can you do it?
All right, good. I'm glad you're thinking. I was gonna say wrong, but I'm glad you're thinking. Okay, so you're saying 138, then you caught yourself, right? And you say, wait a minute, that doesn't make sense. So the degrees of freedom is the total number of possible outcomes minus 1. So how many possible outcomes do you have? Not the total number of children, right? The total number of children is 500. So how many total possibilities are there? 3. Very good, Amar. And there's 3. What are the 3? All right, that's right. Good. Big S, or big S, or little s. There's only 3 possibilities.
Minus 2.
So it's 3 minus 1 equals 2. So that is your degrees of freedom. 2. So you know your p-value. That's 0.05 because that's what we normally use. If they give you a different p-value, then you're going to have to use a different p-value. And now you know your degrees of freedom, which is what? 2. So the 2 tells you the row. You see the row? That's your 2. Your column is what? 0.05. Be careful, because there's a difference. You see 0.5 and 0.05. So what's your critical value? What is the critical value? Your degrees of freedom. Read the chart, guys. There's a chart. 2 is your degrees of freedom. Your p-value is 0.05. What's your critical value? 5.991. I can tell you right now, a lot of people keep saying 1.386. Why did they do that? Because you looked at 0.5, you didn't look at 0.05. Yeah, it's easy to make them. That's why I wanted you to think first. Because you need to, I'm glad you made the mistake now, right? Because when you're on a quiz or a test, you don't want to make that easy mistake. It's easy to make. You're moving through those numbers real quick, and you're just going to pick, so you have to check and double check and check yourself again. So your critical value then is 5.991. That's your critical value. Yes, question in the back?
If they usually is 0.05, how did it become 5.991?
This is a chart. It's not a formula, right? So you don't know what the relationship is between 0.05 and the degrees of freedom, do you? Nobody told you that, did they? We got a chart. You read the chart. That's where we got 5.991. Look at it. I looked at 0.05. I went down to 2. Why did I go to 2? Because that's my degrees of freedom. Why is it 0.05? Because I said that's what we're going to use. So I go down, find the second one. What's there? What's in that cell? What's in that cell? What's in that cell? I go to row 2 because that's the degrees of, and I go to 0.05, and where those two meet, there's a cell. What's in that cell? 5.991. That's how I found that. No, you don't calculate that number. You just find it on the chart. Are we clear?
Yes.
So that is your critical value. Think about what that number is really fantastic. It's really important. Think about what that number means. Think about what it means. That means that any chi-square, any test, and this is what we're talking about, chi-square. When we calculate our chi-square, if that chi-square is bigger than this critical value, where is it going to be? To the right. And therefore, it's going to be what? In the rejection area. So what are we going to do with that number? What are we going to do with the hypothesis, the null hypothesis? We're going to reject it. We reject the null hypothesis if your chi-square value is bigger than your critical value. And that's the only reason we find this number. So we can compare our chi-square to this. Now calculating chi-square is easy. You just need a calculator though. I would definitely have a calculator. Question? Yeah, go. So chi-square, it's like this. It's chi and then square. It's actually not squared. It's not a variable square. Don't try to take a square root. Write that down. Don't try to take a square root of it. It looks like this. It's the Greek letter chi, and they generally put a 2 on there. But that's not x squared. That is not x squared. That is not x squared. Don't try to do some fancy stuff and take a square root of something to get rid of it and just get x. That's not how it's going to work, okay? So chi-squared is equal to what? It's equal to another fancy letter. It's actually a symbol, right? At least a letter. A letter in the Greek alphabet that means that's what? What is this? Somebody know that symbol? It does mean some, but anybody know what it is? It's not an E, it's epsilon. Epsilon. Yeah, it's pretty much where we get our E, but epsilon is a Greek letter. E is an English letter. All right, it does mean sum. It means the sum of whatever comes after it. So all the different values after the sum of what? The sum of this equation. You ready? It's going to be the observed minus the expected, square that, and divide it by the number of expected. For what? For whatever that particular in this case, genotype is, right? So you do that for big A, and then you do that for what? You do it for what then? You do big A, and then you do it for what? Big A, little a. You do the same thing. Observed minus expected, square that, divided by the expected, plus whatever you get for little a, which is, again, observed minus expected, square that, divided by the expected, right? That's your 3, right? You got 3. You got 3 this time. Sometimes there'll be 10 of them you gotta do, right? Add all that up, that's what this sum means, you see. This is telling you to take all of these and do what with them? Add them, right? The sum. Yes?
On the test, are you gonna have the formulas on it?
I kind of don't want to tell you the formula for chi-squared on the quiz, but I will. The reason I don't really want to is because you should be able to do that. It's not, the formula's not that hard, but I'll give you the formula. And the hard part's going to be to figure out where to go. I'm not trying to trick you, I just want you to work at learning that formula, okay? But good question, it's a really good question. All right, so somebody do the math on this? I already did the math, I know what it is, but I want to see, can you all get your calculators out and start doing them? Start doing the math. What would be my observed for big A, big A? 139 minus what? 125. Then that squared, whatever that answer is, square it. And divide by what? 125. Do it. Get the calculus out and do it.
So what goes in here?
What is the type? What's
It
it?
say, is that an O or a 0?
That's
O
an
to
O.
E, O to E. Observed and expected.
Yeah. See, I know you guys have all the answers. You do. You got them in that head of yours. You just got what you're feeling is panic, and you're letting the panic.
Expected.
That's right. Observe minus. You see, that's where you're getting this. See, you got it. What am I doing? Look up here. Look at where it is. Observe minus expected. That's why they had you put it in the chart, right? Because if you have that number, then all you can do is square it, divide it by the expected, and you're done. That's why you need a scientific calculator. Your phones don't work that well for this. Why do your phones not work that well? Because those phones, no matter, and I have an iPhone too, and it just doesn't work as well as a good scientific calculator. You can do it so much faster with a good scientific calculator. I got a scientific calculator. I got a graphing scientific calculator on my iPhone. I'm telling you, there's nothing better than a good calculator that can do that. By the way, when I was a kid, we had to do this by hand. You're lucky. I had to memorize square root tables. We had a slide rule. You guys complain about stuff. Sometimes I roll my eyes when in my head. I don't want to insult you, but you guys complain about stuff, and I just want to say, "Oh, you have no idea." Although I feel bad for you because you got a lot of stuff to learn. We had less stuff. We had to memorize more detail. We had to get more detail done. You guys are more broad. You just don't. You guys got tools like the internet and calculators. I didn't have a calculator. My father got one of the first calculators ever made. Hand calculators for the. Is it HP? Cost him $100. It couldn't do half of what your calculators can do. Didn't graph or nothing. Had little tiny light bulbs. Little tiny light bulbs for the numbers. Fancy. Not one of the first calculators, but one of the first handheld calculators they put out. There were calculators a long time ago. All right, are we done? We got the number? Somebody give me a number.
No.
What number did you get?
For the first one, I got 1.5.
All right, so for the first one, that's A, big A, big A. You got what? 1.5, 1.568. For the next one, you got what? I think that's close, if it's not absolutely right.
Yeah. The next one is 3.364.
And the next one?
Yeah, I ain't get there yet.
14.792. Now take those three numbers, add them together, what do you get? Remember, chi-square is just adding. What is it? These three numbers added together, what is it? I think that's a rounding, you're rounding.
No,
Is
it
it?
is 20. It is 20.
Okay, so you're saying 20, period. So what number did you get? Yeah, that's the number I got. 19 point what? 724? That sounds more like it. But honestly, 19.72, as long as you're doing the math right, and there's some minor rounding errors, I doubt that anyone's ever gonna give you a problem where it's gonna be a difference between point something, right? So now we found chi-square. This is your test, you see? This was your test. This is what you're gonna use to test whether your observed values are not random, that there's something else going on. And this value here is your chi-square, you see? So what are you going to compare this number to? Somebody said, what? Your critical value. And what are you going to compare? So is this value bigger or smaller than your critical value? It's bigger. So it's going to be to the right or to the left of your critical value?
Right.
To the right. When you're reading a graph, are you going to put that, your critical value is here at 5.991, your number's 19.724, it's somewhere way over here, right?
Yeah.
So what area is it in? Rejection area. So if your chi-square value is bigger than your critical value, you do what? You reject what? The null hypothesis, which means what? Is it random? No, it's not random. So something else is going on. In this case, natural selection. Natural selection is going on. All that work to find that out. So you got to get this done. You have number 11. You have to finish answering the questions right now before you go. By tomorrow, I expect number 12 done. I expect pages 1 through 8 are complete and perfect. And 1 through 8 should be quick for you. Right? If it's not quick, if the answers aren't coming quickly and you don't feel confident in your answers, guess what you should do? See me. See me. We're running out of time. We got till Monday. So you need to see me. I'll be here Saturday school too. I'll be here tomorrow after school and Saturday school. See me. Make sure that you understand 1 through 8 for sure. And don't forget, that's still not even discussing Hardy-Weinberg, right? Don't forget that p squared plus 2pq plus q squared equals 1. Don't forget p plus q equals 1. Don't forget what is the probability. How do you take how many you have divided by the total to get your percentage? How do you calculate? Someone actually said over an email that they didn't understand, that they felt kind of cheated because they didn't understand how to find a percentage if you're given a decimal, or decimal if you're given a percentage. You should know that. I went over it in class, right? If you have a decimal, you want the percentage, you move the decimal what? Twice to the right to get the percentage. If you have the percentage, we're going to move it to what? Twice to the left. Do the math. If you're confused, use a calculator. Right? But remember, in some AP courses, you're not allowed to use calculators, so you should be able to do this kind of simple math. All right, that's it. That's everything. I'll upload this when I get home, and everybody will have a copy. Wrap up. It's 3:13. Finish answering these questions now before you go.